What's up mga genetics? At ngayong araw ay tuturo ko sa inyo ang tungkol sa... Wait lang. Nakikita nyo ba? Siyempre, lapit ko na lang. Hindi nyo makikita eh, layo eh. Okay. Pangit ang sulat ko. Ayan. Ito na talakayin natin ngayon. Gene function. So ayan no, gene function, protein and enzyme. Eraser ko. No? Gene function, protein and enzyme. Tapos, may dalawa pa akong tatalakayin mo dito. Under ito ng gene function, protein and enzyme. Lang. Yung isa, basahin ko na lang kasi pag sinulat ko pa matagalan eh. Gene-enzyme relationship, tsaka yung pangalawa, yung inborn errors of metabolism. So, ngayon, tuturo ko sa inyo kung paano nangyayari. So, ayun. Sa genetic control of proteins, ito yung gene-enzyme relationship saka inborn errors of metabolism. Bago natin talakayin itong dalawang to, ano nga ba yung naiisip nyo kapag sinasabing yung salitang protein? Protein muna. Ano naiisip natin, di ba? Ikaw? Galing! Hindi, syempre, ang protein, una natin narinig sa mga salita sa pagkain, di ba? Sa karne. Tapos minsan, yung protein, pag sa hindi ka naman ganun kaano sa genetics, sa biology class, maririnig mo yung protein as parang parang nakaka-relate siya sa amino acid. So, ayun. Pero bago ko i-discuss, sabihin ko yung mga totoong definition yan. Yung isa naman, yung enzyme. Kailangan malaman natin yung mga term na to bago natin sila pag-aralan. Para mas madali natin maintindihan. Ano yung enzyme? Kasi usually di ba may makita pag sa enzyme, mga stem cell. Pero mukhang malayo siya dun eh. Tapos yung pangalawa, ay pangatlo yung metabolism. Kailangan natin malaman yung mga word na to. So ayun guys. Ano yung protein? Ang protein, ito yung linear chain of amino acid. Then, yung formula ng amino acid is NH3 minus di ko alam yung tawag ito, CX minus COOH. Ngayon, yung amino acid meron siyang 20 properties. Kung babanggitin ko pa, sige, isa-isahin ko. Ay, by the way guys, yung ano pala, yung pag isusulat natin yung code ng amino acid, lagi lang siyang 3-letter code. Like for example, yung alanine ay alanine, ALA lang. Pag isusulat mo yung pagpapaiklin mo. So, ang 20 na yon ay yung alanine, arginine, aspartate, cytine, glutamate, glutamine, glycine, histidine, isoleucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine. Bali lahat yun, i-propose ko lang dyan sa screen, makikita niyo yung table. So ayan, ang tungkol natin ngayong araw sa intro to protein and enzymes ay yung basic protein structure, life of protein, tapos yung sa enzyme, yung theory of enzyme. So ayun guys, reminder lang tsaka importante to. Not all enzymes are protein and not all protein are enzymes. Kasi minsan akala ng iba pag sinabing protein, enzymes yan. Pag sinabi enzymes, protein yan. Kasi may iba-iba silang function. Doon ko explain So ayun nga guys, sa enzyme, mangaral natin yung theory niya ng enzyme function. Tapos hindi lahat ng enzyme ay protein and vice versa. Tapos yung enzyme regulation, mahalaga rin yun. Tapos, syempre nabanggit ko na rin sa inyo kung ano yung protein. So bakit nga ba mahalaga yung protein? Why protein is important, my friend? Bakit nga ba siya mahalaga? Bakit? Kasi nga yung protein, ito yung catalyst ng cell. Siya yung responsable sa metabolism. Yung metabolism, di pa natin siya nababanggit ko ano siya, pero explain ko sa inyo kung bakit mahalaga yun mamaya. Isa pang ginagawa ng protein, ito yung interconvert ng energy. Siya yung nagko-convert ng energy, syempre. Tsaka yung protein, siya rin yung nagpe-permit ng selected import and export ng mga molecules mula sa cell at saka sa organelles. And then, yung protein, ito yung nag-aalaw ng paggalaw, ng movement. And then yung protein, ito yung nagpo-provide ng structural support sa organism, tsaka sa individual cells nito. Yun nga. Yun guys. Unat naman ako. So ayun nga guys, sa enzyme, mangaral natin yung theory niya ng enzyme function. Tapos hindi lahat ng enzyme ay protein and vice versa. Tapos yung enzyme regulation, mahalaga rin yun. So kung ano nabanggit ko kanina, yung protein ay meron siyang 4 na hierarchy structure. Yung una is yung primary structure, which is yung sequence. Pangalawa, yung secondary structure, yung mga alpha helix. Explain ko ito mamaya lahat, isa-isain ko. Alpha helix, beta strand, tsaka yung random coil or yung turn. Tapos yung pangatlo, tertiary structure. Ito yung beta barrel, yung ARM repeats, tsaka helix loop, etc. Yung pang-apat, o minsan, sometimes, ito yung quaternary structure, yung protein to protein interaction. So ayun, sa primary structure nangyayari yung sa amino acid yung peptide bond formation. Ayun, sa primary structure nangyayari sa amino acid yung tinatawag natin na peptide bond formation. Yung peptide bond formation, ito yung condension reaction
of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block, of a protein. They are held together by peptide bonds. In protein synthesis, amino acids are added to form a polypeptide chain, and proteins are made of one or more of these polypeptide chains. Genes, which are made of DNA, determine the order and number of these amino acids. That sequence is critical to the protein's structure and function. In our mutations video, we talk about how one amino acid can be changed in sickle cell disease. Even a single change of an amino acid has the potential to affect a protein's function. We do want to point out each amino acid has a carboxyl group, an amino group, and an R group. An R group is also called a side chain. So even though we have them drawn here like a chain of circles, realize that each of these circles we're drawing is an amino acid, like...
So, sa secondary structure naman, sa secondary level, yung sa next level ng folding, yung R group interaction, sobrang halaga nito. Dahil may tinatawag doon na hydrogen bond. Itong hydrogen bonds yung nagde-dictate ng secondary structure. Isa pang kinahalaga nito ng hydrogen bond, sila rin yung nagko-connect sa beta. Sa beta strands. Tsaka, pero ang pinakamahalaga talaga dito, yung tinatawag nila na hindi ko siya gano'ng magets, pero yung may tinatawag silang alpha helix. May papanood ako sa inyong video about that. Para mas magets niyo yung report ko. Alpha helix. Ngayon, alpha helix. Ito alpha helix ito, may tatlo siyang representation. Yung una, basta may makikita ka yung diagram. Mamaya. Yung una is yung ball and stick. Kasunod yung backbone. Then lastly yung ribbon.
In secondary structure, the sequence of amino acids that we mentioned in primary structure can fold in different ways. The most common ways are the alpha helix and the beta pleated sheet. And which one of those foldings the protein does depends on the amino acid arrangement it has. Both of these shapes are due largely in part to hydrogen bonds. Those hydrogen bonds can occur at specific areas of the protein's amino acids. Specifically, these are hydrogen bonds involving the backbone of the amino acid structure. On to
laman ng structure. Dito na nagkaroon yung beta strain na gumagawa siya ng plated sheets. At saka nangyayari rin dito yung helix tsaka strand, gumagawa na sila ng fold.
tertiary structure. This is looking at more folding that occurs in the 3D shape of a functional protein, and a lot of this is due to something we haven't mentioned much, the R groups, also called side chains. See, the amino group and the carboxyl group are generally standard parts of an amino acid, although the R group found in amino acids can vary among different amino acids. That means the R group can define the amino acid and can make amino acids behave a certain way. For example, some R groups are hydrophilic. They like water. Some R groups are hydrophobic. They don't. And remember that proteins contain many amino acids, which can contain different R groups, and so different areas of the protein can therefore be impacted based on those R groups. When protein folding is going on, amino acids with hydrophilic R groups may hang out on the outside, while hydrophobic R groups, where are they? They might hang out at the inside part of the protein. The 3D shape is due to other interactions besides hydrophobic interactions. Ionic bonds, van der Waals interactions, disulfide bonds, and hydrogen bonds, all involving the R groups, can influence the folding occurring in tertiary structure. Something to explore. Now, when we've been talking about a protein, we've been talking about a polypeptide chain that has been folded into a functional protein. But proteins can be made of one or more polypeptide chains. And
Sa huling structure, yung quaternary, ito naman nagbobo ng hemoglobin. Ito yung nabobo yung deoxy.
each of these polypeptide chains can be a subunit, and interactions between them, such as hydrogen bonds or disulfide bonds, can keep them together. Going back to folding, I know what you might be thinking. Who is doing this folding anyway? Are the proteins just folding themselves? Well, the interactions mentioned, like hydrogen bonds and R group interactions, are occurring depending on the protein's own amino acids. One reason why amino acid sequences are very important for protein function. But folding is far more complex than that, and there can be intermediate steps involved when a protein is folding. In fact, there's a phrase you can search called the protein folding problem to learn more about the questions scientists continue to explore regarding protein folding. Research has shown that proteins often have help in the folding process. Chaperonins, for example, are proteins that can help with the folding process. They have almost a barrel shape. Proteins go into them, and the chaperonin tends to have an environment that is ideal for the protein's folding, and this can help the protein be folded correctly so it's functional. Just wish I had something like that for my towels. All of these interactions we mentioned in primary, secondary, tertiary, and quaternary structure are paramount for a mature protein to have its correct shape so it can carry out its function. And that's very relevant. There are many diseases that are related to protein misfoldings. Check out some of our further reading suggestions in the description about that. One last thing we haven't mentioned. Each protein has an ideal environment for functioning, which might include a certain temperature or pH range. If the protein is exposed to something outside of its ideal temperature or pH range, exposed to high heat, for example, you can disrupt the interactions that we had talked about taking place at the different structural levels. This can denature the protein, which disrupts its shape. This prevents it from functioning correctly, and depending on what caused it to be denatured, sometimes you are interfering with many levels of protein structure. Sometimes it's just one or two levels. Sometimes denaturing a protein may be reversible, but in many other cases, it's not. The environment that a protein is in definitely matters for its functioning. Well, that's it for the Amoeba Sisters. Is it odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are we're much, much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least to us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site, where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. And something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real-life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. Now, with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down proteins. So, as you can see, the digestive system is very involved with enzymes. Another thing to point out is that enzymes don't always work alone. Sometimes they get some help. Some often underappreciated but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac-Man analogy yet, right? In the game Pac-Man, there are these ghosts. And when they touch the Pac-Man, it makes this sound. It's like, no, 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 no. And the Pac-Man shape gets all distorted in the process. So what does this have to do with enzymes? No, there aren't ghosts around, but enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes. Keep in mind that if you have an interest in this topic, many medical researchers have a large focus on enzymes. Enzymes regulate a lot of the body processes, and many diseases can involve specific enzyme production, or the lack of it. Well, that's it for the Amoeba Sisters, and